on Wednesday we left off talking about what happens when oxygen is not available to be that terminal electron receptor in the electron transport chain. If you recall, without oxygen to draw those electrons through, there are traffic jams, and those traffic jams prevent ATP from being created in abundance using NADH and FADH2 as the electron carriers. So we looked at a couple of examples of what organisms can do in order to produce ATP in the absence of oxygen. We saw that using the process of glycolysis, organisms can produce ATP, but in order to recreate that NAD+, which is necessary to carry those electrons in order to produce that ATP in glycolysis, there's a secondary compound that is produced. So lactic acid in this case is produced by lactate dehydrogenase, and by producing this organic compound, this allows the regeneration of NAD+, so the cycle can continue. And this is what happens in your muscle cells when lactic acid builds up when you're not getting enough oxygen and you're exercising really hard. This is showing fermentation on the right side, specifically ethanol fermentation. So same idea applies. In this case, glycolysis is occurring and NAD+ is becoming reduced to NADH. This is allowing ATP to be formed. In this case, one is broken down into pyruvate, 2 CO2s are given off, and pyruvate decarboxylase is going to break that down further, and alcohol dehydrogenase is going to break down that 2-acetaldehyde into ethanol as a byproduct, and that ethanol is again produced so that the NAD+ is regenerated to start this process again. So the CO2 that is given off during fermentation, this is where the bubbles come from in beer and other alcoholic beverages such as champagne. So I'll show you another animation here that talks a little bit about NAD+ being regenerated.
All cells are able to synthesize ATP via the process of glycolysis. In many cells, if oxygen is not present, pyruvate is metabolized in a process called fermentation. By oxidizing the NADH produced in glycolysis, fermentation regenerates NAD+, which can take part in glycolysis once again to produce more ATP. The net energy gain in fermentation is 2 ATP molecules per molecule of glucose. Fermentation complements glycolysis and makes it possible for ATP to be continually produced in the absence of oxygen. There are two types of fermentation. Alcohol fermentation, which occurs in yeast, results in the production of ethanol and carbon dioxide. Lactic acid fermentation, which occurs in muscle, results in the production of lactate.
Alright, so to reinforce this, animals take pyruvate and they reduce it into lactic acid, also known as lactate. Fungi, such as yeast, the pyruvate is converted into acetaldehyde, CO2 is given off, and then acetaldehyde is reduced further into ethanol. Okay, but it's important to understand that you and I aren't just drinking sugar solution, right? We are breaking down macromolecules, and we talked about the different types of macromolecules in chapters 2 and 3. So let's look at what happens to these various types of macromolecules and where they enter these different parts of cellular respiration. So the non-glucose carbohydrates are converted into glucose before they're processed. So lipids, proteins, nucleic acids, all of these metabolic pathways interact with the carbohydrate metabolism pathway, and most of them are reversible. So glucose molecules can be produced through gluconeogenesis as well. And I'm not going to make you guys memorize this chart. I just want you to see that it doesn't always just start with glucose, because you and I do not consume sugar solution, right? You can kind of see how glycerol might enter a little bit later than polysaccharides. Fatty acids may be able to enter just before the citric acid cycle. We may have some amino acids that can enter in the citric acid cycle. And notice that it goes in both directions. So this is a highly complex process, And we do follow glucose through the process of cellular respiration because it is a very fast sugar. It provides us with fast energy and is therefore much easier to follow through all steps of this process. Okay, so if we look at fat in particular, we know that triglycerides are broken down to fatty acids and glycerol because those are the different portions that make up their structure. And then glycerol is converted into an intermediate in glycolysis. The fatty acids are cut apart at 2 carbons at a time, and they are converted into acetyl-CoA, and we call this process beta oxidation. So recall that acetyl-CoA, this is what goes into the citric acid or Krebs cycle. All right, so if you need to review these concepts again, you can watch the summary video on cellular respiration. Recall that there are a couple values that are different, so do defer to the textbook and to your cellular respiration homework, which hopefully you've completed for Friday, February 26th. But we're going to move on now, and we're going to talk about photosynthesis. So how is it that the food that we eat is actually produced? Like, where does that sugar come from? And we're going to watch a little video overview on photosynthesis.
In order for plants to grow, they need inputs of carbon dioxide, water, and energy. The chemical process by which plants use these resources to manufacture glucose, the building blocks of plants, is called photosynthesis. In the process, oxygen gas is produced as a byproduct. The energy for photosynthesis originates in the sun and arrives at the earth as sunlight. This light has both a wave and a particle nature. The particles, or photons, are the smallest units of light. Photons oscillate along a path, which is measured as wavelengths. The light emitted from the sun contains photons in a wide spectrum of wavelengths, called the electromagnetic spectrum. Photosynthetic organisms use only a small portion of the electromagnetic spectrum, called visible light. Photosynthetic organisms contain pigments that facilitate the capture of wavelengths of light in the visible light range. The color of the pigment comes from the wavelengths of light reflected. Plants appear green because they reflect yellow and green wavelengths of light. Red and blue wavelengths of light are absorbed by these pigments and provide the energy that is used for photosynthesis. Within eukaryotic photosynthetic organisms, also known as photoautotrophs, the chemical reactions of photosynthesis occur within plant cells in specialized structures known as chloroplasts. Photosynthesis consists of 2 sets of reactions, the light-dependent reactions and the Calvin cycle. Within the chloroplast are small disc-like structures called thylakoids, which are surrounded by a fluid-filled space called the stroma. The reactions that synthesize glucose, the Calvin cycle, occur in the stroma. The light-dependent reactions occur in the thylakoid. It is here that conversion of light energy to chemical energy is initiated. In most photosynthetic organisms, thylakoids contain pairs of photosystems, called photosystem 1 and photosystem 2, that work in tandem to produce the energy that will later be used in the stroma to manufacture sugars. The photosystems of the thylakoid consist of a network of accessory pigment molecules and chlorophyll, the molecules that absorb the photons of light. Within the pigment molecules, the absorbed light energy excites electrons to a higher state. Photosystems will channel the excitation energy gathered by the pigment molecules to a reaction center chlorophyll molecule, which will then pass the electrons to a series of proteins located on the thylakoid membrane. Photons of light strike photosystems 1 and 2 simultaneously. We will examine what happens with the photons striking photosystem 2 first. The energized electrons are passed from the reaction center of photosystem 2 to an electron transport chain. The electrons lost by photosystem 2 are replaced by a process called photolysis, which involves the oxidation of a water molecule, producing free electrons and oxygen gas. While this oxygen gas is a byproduct of photosynthesis, it is an important input to the cellular respiration pathways. As electrons pass through the electron transport chain, the energy from the electron is used to pump hydrogen ions from the stroma to the thylakoid, creating a concentration gradient. This gradient powers a protein called ATP synthase, which phosphorylates ADP to form ATP. The low-energy electrons leaving photosystem 2 are shuttled to photosystem 1. Within photosystem 1, low-energy electrons are re-energized and are passed through an electron transport chain where they are used to reduce the electron carrier NADP+ to NADPH. When the chloroplast is receiving a steady supply of photons, NADPH and ATP molecules are rapidly being provided to the metabolic pathways in the stroma. Therefore, the ATP and NADPH formed during the light-dependent reactions are used in the stroma to fuel the Calvin cycle reactions. The Calvin cycle consists of a series of reactions that reduce carbon dioxide to produce the carbohydrate glyceraldehyde 3-phosphate. The cycle consists of 3 steps, the first of which is carbon fixation. In this step, carbon dioxide is attached to ribulose 1,5-bisphosphate, resulting in a 6-carbon molecule that splits into 2 3-carbon molecules. The second step is a sequence of reactions using electrons from NADPH and some of the ATP to reduce carbon dioxide. In the final step, ribulose 1,5-bisphosphate is regenerated. For every 3 turns of the cycle, 5 molecules of glyceraldehyde 3-phosphate are used to reform 3 molecules of ribulose 1,5-bisphosphate. The remaining glyceraldehyde 3-phosphate is then used to make glucose, fatty acids, or glycerol. It takes 2 molecules of glyceraldehyde 3-phosphate to make 1 molecule of glucose phosphate. Thus, the Calvin cycle has to run 6 times to produce 1 molecule of glucose. These molecules can remove their phosphate and add fructose to form sucrose, the molecule plants use to transport carbohydrates throughout their system. Glucose phosphate is also the starting molecule for the synthesis of starch and cellulose. Plants produce sugars to use as storage molecules and structural components for their own benefit. By utilizing the energy of the sun, along with inputs of water and carbon dioxide, plants act as glucose factories. Photosynthetic organisms are the primary producers of glucose on the planet. They also produce oxygen gas as a byproduct, and thus serve as the foundation of life, providing food and oxygen for the complex food webs on both land and in the oceans.
I want to remind you that in addition to watching this video, if you would like to watch the video itself without having to listen to the lecture portion, you can go into each PowerPoint, and anytime something is underlined with blue or a picture is outlined with red, what that means is that there's an embedded video link. So put it into presentation mode, and then you can just click on it and it will automatically open up the web browser. So if you recall, we talked about the reaction of photosynthesis before as the opposite of cellular respiration. So photosynthesis is taking in carbon dioxides that were given off during cellular respiration. They're also taking in water, and using the energy of the sun, they're converting that into glucose, which is C6H12O6, and they're releasing oxygen for us to breathe in. So basically this is a balanced or coupled reaction, because we can look at how cellular respiration here is the exact opposite. So taking that sugar that the plants have made and breathing in the oxygen that the plants have produced to yield carbon dioxide and water, which will in turn be taken in by the plants in order to produce sugars. So if we look at how photosynthesis works, there are two major types of reactions that occur. There are the light reactions, and this is when light energy is captured using electrons, and they're used to create ATP and NADPH. So NADPH is very much like NADH in cellular respiration. It is an electron carrier. Then there are the carbon fixation reactions. Some books will call them the dark reactions or the light independent reactions. But what they do is that the ATP and NADPH, they're used to build carbohydrates by linking together those carbon dioxide molecules. But let's take a step back, because in order to talk about how photosynthesis works, we need to talk about how light works. And light is actually a type of electromagnetic radiation. And visible light is between the wavelengths of 400 and 700 nanometer. So what a wavelength is it's the distance between the peaks of two waves right here. Okay, so it's the distance from one peak to the next on a light wave. So the shorter the light wavelengths, the more energy that is contained in those wavelengths. And the longer, the less energy. And we can look to see that in visible light, at each of those different classes of wavelengths, they correspond to different colors in the visual spectrum. So if you had a prism, which is clear and usually made out of glass, it can bend the light and it can actually show all these different colors. So that's why you get rainbows. But the real question is, how do plants actually get the energy out of light? So we have to look at what happens when light strikes a molecule. So here we have a photon. So light is simultaneously a particle and a wave. So this photon is traveling until it hits a molecule, and that molecule could absorb the energy from that photon and basically leap an electron up to a higher energy level. So this is looking at the ground state here. When the photon hits that molecule, it can move that electron to the higher energy level. Okay. But light can also scatter off, or it can reflect. And if you've ever wondered why plants are green because the one part of the spectrum that they do not absorb is actually green light. So when we are looking at how light is absorbed again, it's because photons are literally knocking those electrons up into a higher energy level. And this is because electrons are prone to be lost. They are orbiting around the center of the atom. And there are some main pigments that absorb light in plants, and these are chlorophyll A and chlorophyll B. So they absorb in the red and blue light spectrum, but not green. Because remember that green is reflected, it's not absorbed, and that's why plants are green. So these activated electrons that are capable of absorbing this energy, they are what power photosynthesis. And what this is showing here, this is showing the absorbance or the activity, so how much of that energy those plants or pigments are getting at these different wavelengths. So what we're seeing is that there are two peaks here. So chlorophyll A is absorbing very well in the blue and the red wavelengths, but almost nothing in the other wavelengths. Another plant here is showing also a peak absorbance in the blue, but also into the ultraviolet a little bit, and then the other peak being more towards the red. But notice how this particular plant is able to absorb a little bit more of the other wavelengths. So different plants have different pigments, and they're able to absorb different wavelengths. Alright, so exactly how does chlorophyll work? First of all, it's housed in chloroplasts. And we talked about chloroplasts as being the organelles that arose from endosymbiosis. They came from a primitive eukaryotic cell or a prokaryotic cell on the way to becoming a eukaryotic cell that consumed a cyanobacterium that over time became beneficial for that cell to retain, and then through a very long time became a part of that cell. So if we look at that chloroplast, what we'll see is that there are these stacks of poker chips on the inside. And each of those poker chips is known as a thylakoid, and that thylakoid membrane is studded with light harvesting complexes in the form of chlorophyll pigment molecules. So each of these complexes has many of these chlorophyll molecules. Here we can see a light harvesting complex with different chlorophyll molecules. So this is showing a protein embedded in the thylakoid membrane. So what happens is if chlorophyll gets hit by a photon and it gets excited, that means that its electron is going to get shifted up into the higher energy level. And then this electron is going to be captured by the next chlorophyll. And eventually it's going to arrive at something known as the reaction center. And that reaction center is going to carry that energy to the next step. But here we can see the different chlorophyll molecules, and they can actually transfer energy amongst one another. All right, and here we can see what is known in general as the photosynthetic Z scheme, mostly because it looks like a Z. Right. So we're looking at photosystem 2 and photosystem 1. So photosystem 2 and photosystem 1 differ in a couple of different ways. First of all, the reaction center chlorophyll, the one that is actually going to send its electron at the higher energy level to the electron transport chain, this has chlorophyll P680. Okay, so this absorbs light at a peak of 680 nanometers. Now P700, this has a peak absorbance at 700 nanometers. But I want you to see what's happening at these different photosystems. In photosystem 2, you have a photon hitting those chlorophyll molecules and being transferred from chlorophyll to chlorophyll until they hit that reaction center. And at this point, water is actually oxidized. So an electron is able to replace the electron that gets knocked up into the higher energy level. And in doing so, that water gets broken down into oxygen and hydrogen ions. So these electrons are then going to be transported through the electron transport chain, and ATP is going to be produced in a very similar way to the chemiosmosis that we saw in cellular respiration. Now photosystem 1, this electron is going to travel to that P700. A photon will hit that P700 and knock that electron up into a higher energy level, and then the electrons will go through electron carriers, and finally NADP plus will be reduced to NADPH. Okay, so that electron carrier is going to be reduced to form that NADPH, which will carry that electron further. So let's watch a short animation by McGraw-Hill that explains this process in video form.
In bacterial photosynthesis, a single photosystem is involved. When an electron is energized by absorption of light, it is ejected from the photosystem reaction center. The electron then passes down through an electron transport system and finally back to the reaction center. The energy released during this electron transport is used to produce ATP. Since the excited electron returns to the reaction center, this mechanism for making ATP is called cyclic photophosphorylation. No reducing power needed for biosynthesis is generated in this process. Plants and cyanobacteria utilize two photosystems which work sequentially to produce both energy and reducing power. First, a photon of light ejects a high-energy electron from photosystem 2. The electron lost from photosystem 2 does not return to photosystem 2, but is replaced by an electron generated from the enzymatic splitting of water and the release of oxygen. The electron then travels from the excited reaction center of photosystem 2 down an electron transport chain and finally to the reaction center of photosystem 1. This electron transport system generates a proton motive force that is used to produce ATP. Since the excited electron does not return to photosystem 2, this mechanism for making ATP is called non-cyclic photophosphorylation. When photosystem 1 absorbs a photon of light, it ejects a high-energy electron, which is used to drive the formation of reducing power in the form of NADPH. The ejected electron is replaced by an electron from photosystem 2.
So the video mentioned this idea of cyclic versus non-cyclic photophosphorylation. And really what they're talking about with photophosphorylation is using light to put that phosphate on an ATP. And the reason that the photosystems are called photosystem 1 and photosystem 2 is that photosystem 1 was the first one to evolve in primitive photosynthetic bacteria, whereas the non-cyclic photophosphorylation utilizes both photosystem 2, which is the second one to evolve, and photosystem 1 in this photosynthetic Z scheme that we're seeing. But let's look at these in words. So if we explain what happens in photosystem 2, this is when photons excite the chlorophyll in photosystem 2 in its reaction center, and it gives up an energetic electron. So that electron is knocked up into a higher energy level. The chlorophyll has to replace this electron by oxidizing water or breaking water down into oxygen and hydrogen and then electrons. This energetic electron then goes through a series of electron carriers, very much like the electron carriers that we saw in the mitochondrial membrane during cellular respiration. And very much like them, they're pumping protons. So they're pumping hydrogen ions, in this case, into the thylakoids. So they're building up those hydrogen ions so that they can flow through ATP synthase in the same way that they did in cellular respiration. So this allows chemiosmosis and ATP synthesis. So after the electron loses its energy, it's ultimately delivered to photosystem 1. And if we explain photosystem 1, this is when the excited chlorophyll in the photosystem 1 reaction center gives up its energetic electron. The chlorophyll is then replaced, the electron, from photosystem 2. So that electron that moves out of photosystem 2 and goes through the electron carriers replaces the electron that is knocked into the higher energy level by light in photosystem 1. And this electron can do a couple different things. So it can be used to reduce NADP plus into NADPH, so it can be making NADPH, or it could be passed through the electron transporters and pump protons. And we'll talk about this in a minute. And eventually it's going to make ATP just like photosystem 2. And we call this cyclic electron transport because ultimately that electron is returned back to its chlorophyll. So you can see how these processes are linked together, and really if you think about it, for a plant, producing ATP is not the ultimate goal. The ultimate goal is to produce sugars for storage and structural components. But if you're looking at a photosynthetic bacterium, that photosynthetic bacterium, its ultimate goal is likely to produce ATP, and that's why photosystem 1 in itself would work. Alright, so let's look at an alternative pathway for photosystem 1. Let's look at cyclic electron transport. So we're going to watch the second half of that video again. So instead of producing NADPH, this energetic electron can go through the pump and basically pump protons into a thylakoid, just like it did in photosystem 2, and have the protons exit through ATP synthase again. So this is that cyclic electron transport or cyclic photophosphorylation.
In bacterial photosynthesis, a single photosystem is involved. When an electron is energized by absorption of light, it is ejected from the photosystem reaction center. The electron then passes down through an electron transport system and finally back to the reaction center. The energy released during this electron transport is used to produce ATP. Since the excited electron returns to the reaction center, this mechanism for making ATP is called cyclic photophosphorylation.
So when bacteria photosynthesize in this way, and they're using photosystem 1, they're not making NADPH. They're only passing those electrons through so that ATP can be made through chemiosmosis in much the same way that ATP is made in the mitochondrion through chemiosmosis as well. And if it were linked to photosystem 2, then NADPH would be made. Okay, so what happens when we have ATP and NADPH? It's time to make some carbohydrates. So we're going to make some sugars. And this happens with the Calvin cycle. Sometimes you may hear this referred to as the Calvin-Benson cycle. And it uses coenzymes ATP and NADPH, and it does this to generate carbohydrate molecules. And there are 3 phases. The first phase is fixation of CO2, and what that means is the plant is taking CO2 in through its leaves, and CO2 is then going to bind to something known as ribulose 1,5-bisphosphate, which we'll be referring to as RUBP. So this is a 5-carbon compound. RUBP is then going to break into 3-phosphoglycerate, or 3-PG, and these molecules consist of 3 carbons apiece. So here you can see the carbon dioxide coming in and binding to RUBP. This is going to, Rubisco is an enzyme that catalyzes this reaction and creates this 6-carbon intermediate that is then split into 2 3-PG molecules. The second phase is when 3-PG is reduced into glyceraldehyde 3-phosphate, which we'll be referring to it as G3P. So this is going to take ATP, so it's going to consume energy from ATP, and it's going to consume energy from NADPH. So where did this ATP and NADPH come from? Do you guys remember? Recall that you have ATP and NADPH. ATP is produced in that electron transport chain that connects photosystem 2 to photosystem 1, and NADPH is a product of photosystem 1. Okay, sugar production is phase 3 and regeneration of RUBP. So what happens to these G3P molecules? 10 out of 12 of them, or 83% or so, are used to produce more RUBP, because this is a cycle, right? And anytime we're talking about a cycle, the first compound in the cycle also has to be the last compound of the cycle. Only 2 out of those 12 molecules, only about 17%, is actually used to make glucose. So if G3P is a 3-carbon molecule, 2 are going to be needed to create 1 glucose. So we need 12 J3P, which is going to require 6 full Calvin cycle turns. So there are 6 CO2 molecules necessary to bind to complete. So it's important to understand that the glucose is not just going to be sugars that the plant can use immediately. They can be bound up in starch as storage, or they can also be structural components like cellulose. So this is what the Calvin cycle looks like. So we have our carbon fixation, reduction in sugar production, and regeneration of RUBP. So I'm going to show you an animation on how the Calvin cycle works.
The Calvin cycle is a series of reactions that results in conversion of carbon dioxide into the organic molecules needed to build new cells. It occurs in the stroma, which is the area of the chloroplast surrounding the thylakoid membrane. During the Calvin cycle, carbon dioxide, CO2, is added to a 5-carbon molecule called RUBP. The resulting 6-carbon molecule is unstable and quickly splits into 2 3-carbon molecules called 3-phosphoglycerate. Using energy from ATP and reducing power from NADPH, which are products of the light reactions, the pair of 3-phosphoglycerates move through a series of reactions and are converted into 2 molecules of glyceraldehyde 3-phosphate. When several of these glyceraldehyde 3-phosphate molecules have been produced, some combine to make glucose, while others are reused in the Calvin cycle. To generate an entire new glucose molecule, the cycle has to turn several times, because each turn of the cycle adds only 1 carbon atom from each molecule of carbon dioxide.
So now we've looked at the major processes that occur when we break down food in our bodies to create ATP, and when plants take in energy from the sun in order to make sugar. So let's look at some key similarities. First of all, they're the reverse reactions of one another. Both of them include a large biochemical cycle. So cellular respiration has the citric acid cycle, and photosynthesis has the Calvin cycle. Both include an electron transport chain that pumps protons. So in cellular respiration, the electron transport chain is in the mitochondrial membrane. And in photosynthesis, the electron transport chain is in that thylakoid membrane within the chloroplast. Both of them are going to use chemiosmosis to make ATP. So both of them are using proton pumps to create a large concentration of hydrogen ions on one side of a membrane such that they can run through ATP synthase in order to make ATP. And both of them use carriers as coenzymes. So recall that cellular respiration use NADH and FADH2 as electron carriers to bring electrons to the electron transport chain. And in photosynthesis, there's a very similar compound known as NADPH, which brings electrons to the electron transport chain in the thylakoid membrane. Now that we've completed talking about photosynthesis, I want to test your knowledge to see if you really understand the process. And we will discuss both cellular respiration and photosynthesis again when you return from spring break. But I do want you to take some time to think about this process and apply what you've learned. And of course, that means I have an assignment for you. So if you go into Unit 2 Materials and you go into the Chapter 6 folder, what you'll see is that I've made a homework assignment for you, which is due the Monday we come back. So what I want you to do is to complete the missing labels and answer the questions in this PowerPoint file. Okay, so the first thing I'm going to do is I'm going to save the file. And I'm going to open it up, and I'm going to save it as photosynthesis homework _ Drews. Just put your last name in it somewhere so that I know it's you. Alright, so there's a couple things that you need to do. First of all, filling in these areas that are asking you questions. So, my name, Dr. Drews. Okay, where are the photosystems located? Well, I'll give you one answer. Okay, so the photosystems are located in the thylakoid membrane, right? And you can tell me where the thylakoid membrane is located. What organelle is the thylakoid membrane located in? Another thing that you need to do is you need to complete labels that are missing. So, for example, in order to do this, you'll create a text box, and then what you can do is you can make it smaller. So I've made it into, I don't know, font of 10 or so. And obviously we're making something here, right? We're making ATP. So for each thing that should have a label, and you can use your textbook to help you, you can look at the PowerPoint slides, I want you to add in those labels and label pretty much everything that you can. So a really fast way to get those labels is to just click on one of the text box that you've made and then Control-C, Control-V. And you can copy and paste and then put this wherever it is that you need to go, and you can type in that as well. All right? So you're going to go through and answer all of these questions. And number 8, this is one that you really need to watch the video at 5 minutes and 50 seconds, because it explains why the Calvin cycle has to run 6 times. So please complete this assignment. When you finish it, save it. Okay, so I'm going to save it. And then you're going to go in, click on this assignment link, and you're going to browse my computer, and you're going to find that file. All right. I'm just going to cancel that. So I want to wish you all a good spring break. I want you to stay safe, and I want you to have a good time as well.